Okay, well, we'll now ask Janet Woodcock, Director of CEDAR, to come up, and it's been, I'm really looking forward to the discussion here.
Thank you, Rob. And good morning, everyone. I've enjoyed all the previous speakers, and actually I think most of their comments can actually be reconciled with one another, believe it or not. I don't really disagree with much of what has been said. The topic of this workshop is incentives. And I think the interest in regulatory use is partly because that's where investment can come. There is very little interest in investment outside of regulatory as far as the new methods and so forth. It's quite interesting, except for PCORI and some other things that are coming along, which is good. But so the question is, what kind of incentivization could regulatory use bring to the use of real-world evidence? So everybody said this. I get it. We all get it. Not only are we limiting development, our practices are not evidence-based in healthcare generally, because we can't generate the evidence to answer the question. We can't tell patients what's going to happen to you or predict what might happen to you. And that's what they want to know. And which medicine should you take, or which course of therapy should you pursue? And it's mainly a gut instinct on the part of clinicians because it's too expensive to generate the answers and so forth. And this is also limiting research and many scientific innovations actually getting into the clinic because it's so expensive, as we just heard. Now, FDA is committed to exploring the use of real-world evidence in regulatory decisions. As Rob said this morning, provisions of the Cures Act require us to do that, so that's in the law, and that's very good. Under the user fee programs that we've agreed to and were recently put into law, we committed to exploration. We're doing a demonstration project called Impact AFib, which you probably heard about already, but it's a educational intervention where we're looking at the outcomes in Sentinel, and this is simply to be a demonstration that this can be done. It's randomized. Recent September 13th workshop with Duke Margolis, which I'm sure you heard about, and then CDRH and CBER just issued guidance for devices. And this is salient to what I'm going to talk about next, but it's about use of real-world evidence to support regulatory decision-making for devices. And it talks about how difficult it is to develop evidence for device development for a variety of reasons. There's a lot of challenges. And they point out that you can not only use real-world evidence perhaps for looking at outcomes, which is mainly what's been talked about here, but also, as we heard from GSK, historical control, concurrent control, natural history, expand a label for an approved device, and safety surveillance are also things that can be used, and their guidance goes through all these. So these, I think, would be incentives for the device industry to help invest in ways to strengthen this science and make it more robust so it can be used. And they also go through the kind of evidence, characteristics of the evidence. They have their own definitions of relevance, reliability, and talking about quality control and so forth in the guidance. So similar considerations apply to use of real-world evidence in drug development, but of course, an investigational product use requires informed consent, as would randomized trials of new uses that use medical records or claims data for outcomes if they're actually a randomized trial. Real-world evidence obviously is used extensively for evaluation of safety of marketed products, and we've heard all morning about some of the challenges there that are, many times the signals we get are not actually borne out after excruciating effort to work on them. But there's very little historical use of the real-world experience in drug regulatory decisions about effectiveness. And obviously that might be the most important if you're talking about incentives, because that's the most expensive part of the whole proposition. there are no hard and fast rules, despite what Dr. Collins said, about how evidence is generated. GCPs or, we can have exceptions to that. With the exception of informed consent and patient privacy in the United States, at least, those are hard and fast rules, more or less, that you have to follow those. And the settings you can do studies in can vary along a spectrum from standard clinical trial setup to a pragmatic trial run in healthcare system. And FDA has accepted trials from all of those. But as I said, it's unusual. But there are trade-offs among data reliability, pragmatism, control of error, safety, and other factors you have to take into account when you decide to do what kind of trial you're going to do. Clearly, for example, you don't want to run a first-in-human trial in a real-world setting, okay? So pragmatically, it's much more appropriate to run that in a very controlled setting. So we're evaluating further the use of real-world evidence. We've done a lot of things. We have approved drugs for rare diseases based on data from registry like case series. I'll talk about a couple of those, or treatment use of a product. We've used registry data as external controls for uncontrolled experience data. We're exploring how randomization would work in registry or healthcare settings, such as our Impact AFib trial. We're collaborating with groups working to improve the validity of key data elements collected in the process of healthcare. And I don't know, Rob, whether that was talked about yesterday or not, but making sure, like what we've discovered, and I know some of the groups working, the cancer groups working on this, found that the healthcare record, the electronic healthcare record, may not have the right stage of the cancer and so forth. So there are all these problems. And at quality by design, if you want to talk about that, which I fully believe in, it would mean you get it right the first time. You don't have to go back and adjudicate that and verify it, or and so forth, curate the data. It should be put in correctly if it's a key element in the electronic health record. And so there are groups working on that. And that's obviously good for health care of the patient, and it also would be good for using those data. And we've spoken to the many groups that are assembling, and we're collaborating with them, that are assembling oncology care data in various ways. And they hope to provide valid platforms for investigations and also use those data. I'd say the cancer field is the furthest advanced for reasons I will speak about in a minute. So here are some types of data we've used. We've used registry data, we've used case series, and we've used treatment data and so forth, where something was used with treatment intent. And as I said, we've also used data from real-world evidence type of data as external controls for rare diseases. So this is feasible and allowable right now. But new science is really means that we're going to have to change our traditions, and that isn't that surprising. There are many more rare and orphan diseases being studied, but even in the common diseases, we're starting to see targeted therapies with companion diagnostics or using next-generation sequencing. Often this narrows your target population down to a very small number of people, and they're scattered throughout the world. They aren't all in one healthcare system or whatever. And so your idea of a traditional trial doesn't work very well. Breakthrough therapies are asking for fast development programs that are efficient. And so we're seeing a lot of different changes in drug development that's driving the need for innovation in how we study these. So how would you use the real-world evidence during drug development? We've already heard about this a bit, okay? But the same is what the Device Center said, natural history information for planning your trial, planning your endpoints. It's critical. And also to use sometimes external controls of different kinds. Biomarker development. Again, neither of these provide the huge incentive, I think, that probably efficacy trials do, but they're still excruciatingly important in success of development programs. evaluate an investigational drug in a hybrid model that can use, say, real-world outcomes. And we have seen this sometimes, where you perhaps do the randomization and the initial assignment in a traditional type of way, and then you measure the outcomes using real-world evidence. Add new indications to an approved drug, and then post-market safety evaluation, which I'm not going to talk about more because I believe Sentinel and related things have been talked about a lot. So what are some of these uses? So real-world evidence is the way we get knowledge that underpins a development program. I mean, what's happening to people now in the last 5 years with this disease? What is their course like? How many outcomes do they have? The only way people get that information is from observational data right now. And the better we can make that, the better you can design the trial. What do people with the disease want to have mitigated? Okay, what is the burden of disease, in other words? That's something often is very poorly understood, and you can use real-world evidence for that. How will endpoints perform? How about patient-reported outcomes? What patient-reported outcomes are used in practice? How are they scored? Are they in the electronic health record, and so forth? And so you really, nowadays, you shouldn't just do some power calculations when you're doing an efficacy trial. You should really do modeling and simulation based on a vast amount of knowledge that you can accumulate from observational data and other trials and figure out what's going on at this point in time if you're doing a trial in that disease. And of course, in rare diseases, you have no idea what's going on. And that's where, and the rarer diseases are where this kind of evidence or evidence from registries can be critical in designing a development program. So burden of disease, rate of progression of symptoms, and so forth. What we've discovered in rare diseases particularly is the experts are usually wrong. They've only seen a couple people, and that rare diseases turn out to be very heterogeneous. And so the developers choose endpoints and time to progression in their studies based on some expert opinion, and it turns out to be just completely off base, and then they have to start over again. So also what we're discovering now is that not only cancer but rare diseases are very heterogeneous, and you get a lot more variability. And here, self-controlled trials may actually be best. Because each person has a slightly different trajectory in their disease, or a large difference in trajectory. If you can use their historical rate of progression and compare it to after the intervention, that is one way of conducting a trial in a rare disease. But what we found is the registries, many of them that we have now, are not very good. And so it doesn't provide the data that you might need to do that kind of study. So biomarker issues, development program may use a predictive biomarker to pick out responsive patients, or prognostic biomarker to pick out people who are likely to have an event, or pharmacodynamic biomarker, to look at whether the drug is working. And usually the data from those needs to come from some observation of their performance prior to initiating the trial of your investigational drug. So understanding biomarkers and their performance in this population, I think, is often a critical part of having a good development program and is used for real-world evidence. So biomarkers, I think, that are critical to a development program. For example, if you're relying upon a prognostic marker to get a certain event rate, you really should have tested that hypothesis using some real-world evidence before you embark upon a $1 billion trial or whatever it's going to cost. So the biomarker, understanding performance of biomarker would be another incentive to use, I think, real-world evidence, and a very good use of real-world evidence. Add a new indication to an approved drug or an extending an indication because the product has been used off-label. Okay, so with ivacaftor, for example, which is a drug for cystic fibrosis, we didn't use these data in the approval of additional mutations, but the Cystic Fibrosis Foundation has a very good registry where they observe the people every quarter and record a lot of information about them. And there were a number of people who were able to talk their insurance company into switching them onto ivacaftor off-label. And so I asked the Cystic Fibrosis Foundation to look before and after at their FEV1s, and if they found that they had a sustained increase in FEV1 after switching over to ivacaftor. Subsequently, we were able to use mechanistic data along with some trial data to add those mutations to the indication. But you could see in the future, you could use this because to add indications to different diseases, especially as we're approving sort of mutation by mutation in these targeted therapies. And that's true with cancer, too. There's a lot of work going on looking at cancer patients who have a driving mutation. It's not indicated in that tumor. Can we collect enough information about their response from real-world evidence to say, yes, those people also respond, and add that tumor, say, to the list? So, and the new indication, if it's a big effect, all right, and that's the really big thing. If you have a big effect and you've had off-label use and you intervene and you see a large difference, then you may well be able to use real-world evidence to add an indication. Now, what about studying investigational drugs? This is the big kahuna, right? Or adding new indications with trials, with randomized trials that incorporate real-world data. Well, I think this, and other people have said this already, Rob has said this, I think this is going to require a hybrid approach. We need to bring the clinical trial universe and the healthcare universe as close together as possible. So as much of the work is done in the actual process of care in the trials to reduce costs and add efficiency. Now, the NIH Collaboratory, I think, is a really good example. And what they found is they have really respectful interaction with the caregivers as partners. They had to do training and learn. They had to get trained themselves, the collaboratory folks, in what caregivers need and what the care process really is like, all right, in order to integrate the trial into an actual care process. But they succeeded, as Rob said, at 1/10 of the cost, perhaps, of traditional trials. There's no reason this type of trial could not be done for regulatory purposes, GCP notwithstanding. All right, because quality by design, if you design how you're going to conduct the protocol and how you're going to manage different errors and which is allowable and which is not, FDA or any other regulator could agree upon that alternative design and agree to accept it. So what I think might be an incentive in this area is the rise of master protocols and what I call trial platforms. What people call them platform trials, but regardless, these are continuous, ongoing trials rather than start and stop trials, all right? And they can study many things. They could study biomarkers, they could study devices, they could study outcomes, right, in a disease. The goal is not to study one single intervention or biomarker. It is to have continuous improvement in the disease outcome. And that is, of course, the goal that we have in the healthcare system is to improve outcomes for people with different diseases. These so far have been run by consortia that have experienced trialists as principal investigators. And as I said, they can evaluate many things over time. And I think the most important thing here is because they are continuous rather than start and stop, they could add elements to incorporate data collection and trial procedures into the care process, and thus have the characteristics of real-world evidence, but continue randomization and certain other things that are necessary, often consent, for studying new products. The advantages of doing these things is that this saves time overall, or there's more investment up front. There's an opportunity to include the community practitioners, better recruitment, better research, and practice integration. We've had a huge, there's another translational problem that people don't talk about, which is we find something in a trial and it's published in a prestigious journal, and yet the community of practice does not adopt it. For years, maybe forever. And if you go out and audit, you find it isn't adopted. And that's because they're off doing practice and other people are doing research, and the two are not integrated in any way. And I think that has to be repaired. Can answer multiple questions instead of one. Yeah, I'm almost done, Rob. The investigators are seen as unbiased toward any given intervention. This would normally study comparative effectiveness, as it's called, and look at the upsides and downsides of one therapy versus another, or one course of treatment. It's patient-centric, and it can use adaptive designs creatively. But the problem is this is novel. It doesn't comport with current model of pharmaceutical development, which is where the funding would come to set these things up generally. It doesn't comport with the academic rewards model, which rewards individual effort rather than collaborative effort. It's harder to set up in the beginning, and it doesn't comport with grant structures as they exist, at least in this country. I think other countries with their health systems are more amenable to this type of approach. So I think as new master protocols or trial platforms are set up, they can be explicitly designed to incorporate real-world data and as extensively as possible, so that makes them less expensive, more feasible. Requires additional work in standardization, data verification, training, and curation. However, it could really pay off. And I think this is one way you can deal with the objections of those who say we need randomization. I think randomization is completely possible, and yet still have most of the elements of the desirable elements of real-world evidence. So in conclusion, I personally believe, like many other people, that the clinical trial system we have right now is, I'll say, broken. It isn't working to meet the objectives for which that we need it for, and that master protocols, trial platforms, and clinical trial networks need to be the future, utilizing healthcare data and actually integrating into healthcare when at all possible. IMI in Europe and groups here in the US are working towards a transformation that will utilize digital health data within a trial system, but there's really a long way to go. Our inability to generate the needed evidence efficiently and in a cost-effective manner will continue to be a major barrier to innovation and to the quality of care around the world. I think regulators are highly willing to make decisions based on real-world evidence, provided this evidence has the characteristics needed for us to rely upon it to make decisions. Thank you very much.